Hello everyone, this is the last in the series of membrane transport. So today's lecture we talk about membrane transport of small molecules across various membranes. So again, remember I had divided this into 6 sections. So today's topic is membrane transport. So the membrane, as you know from other studies in our previous biochemistry classes, has a hydrophobic interior, and that serves as a barrier to passage of most polar substances. So in some ways that's good because the cell does have to maintain intercellular concentrations of solutes that are different from those outside the cell in the extracellular matrix. But you do still have to be able to, in the cell, get things in and out of the cell. So the membrane transport systems are responsible for that. And it is interesting to note that all membrane transport proteins studied so far have been multi-pass transmembrane proteins. So that speaks to the complexity of these membrane transporters and the importance of their role. So here is a slide here that shows the differences in different ion concentrations. And you can see in the cytosol that these concentrations are far different. So for sodium, we have a low concentration inside and a high concentration outside. It's the opposite for potassium. We have a high concentration inside and a low concentration outside. The potassium difference is particularly large because calcium is involved in signaling events. So we have a very low concentration of calcium inside and it's much higher outside. So the transporters help maintain these differences. So I've divided this into sections. I've tried to organize this content. It's a little different because I'm drawing on various textbooks, and different textbook authors have different ways of organizing this content. So this is the way I've collected all of these things and tried to organize it into something that's reasonably coherent. So let's start simply with something called simple diffusion. So here, there's no transport protein at all, and this involves gases and small uncharged molecules only. So in my graph right here, because the membrane has this hydrophobic interior, small uncharged molecules like gases can go across without a transporter, but large uncharged large polar molecules, that's not shown here, it's not really a size thing, it's a polarity thing. So it's so if you have small polar molecules, they can cross, but they cross a lot more slowly. And ions even more slowly. Not only are they charged, but they have that shell of water molecules around them. So if we look at this this permeability index right here. You can see low permeability, we have ions on one side, and then for high permeability, we have nonpolar small gases. So these kinds of molecules can cross the membrane without the aid of a transporter. So here's a chart that I got from Lotish, and this would be in the simple diffusion area right here. So no protein, and because of that, these solutes have to be transported along a gradient, and no ATP hydrolysis is obviously responsible for that. So oxygen and carbon dioxide, many drugs, in fact, that's a way you get drugs into the cell is because of their being nonpolar molecules, steroid hormones. And steroid hormones are fairly large. So again, this is a polar thing and not a size thing. So if you are a nonpolar molecule, you can get across the membrane without the aid of a transporter. So this is from Alberts, and again, it shows a molecule going across a membrane without the aid of a transporter in the direction where you're going along a concentration gradient. So anyway, simple diffusion, no protein transporter is involved. Okay, so now let's go on to a much larger class of molecules where they do need transporter proteins in order to get across. So I've divided these into different areas. So there are 3 classes of transport proteins. So the first one I'm going to talk about are ATP-powered pumps. So the ATP-powered pumps need ATP because this is active transport, and here you're transporting a substance against its concentration gradient. So if we look over here on the right, this blue square right here, if you want to get the blue square to go in this direction, if you're crossing the membrane against its concentration gradient, that's active transport, and ATP hydrolysis will be involved. So that's shown over here too. So if this molecule right here is going from the cytosol to the exterior, and it's going against a concentration gradient, and ATP hydrolysis is involved. Usually what happens is the ATP hydrolysis means that it's not just a hydrolysis like it's shown here, but the phosphate is used to phosphorylate the pump. And that not only maintains the energy of that phosphoanhydrate bond in the ATP, but it also allows a conformational change to occur within the pump. So if I go back to this table, we are here. So active transport requires a protein. The solute is transported against its concentration gradient. It is coupled to ATP hydrolysis because of that. And examples here are ions, small hydrophilic molecules, and lipids. And again, because we're going against a concentration gradient, ATP is necessary in order to do this. So let's put the ATPases into further classes. We have P-type, V-type, F-type, and ABC superfamilies. So let's talk about different examples. And some of these you have seen before, and some of them maybe you haven't. Okay, so let's talk about the P-type ATPases first. So the P-type ATPases involve the phosphorylation of the pump. And that's shown here. The ATP, when it's hydrolyzed, the phosphate is used to phosphorylate the pump. So that's why these are called P-type ATPases. And here are some examples of P-type ATPases. So the sodium-potassium ATPase you've seen before. The calcium ATPases, they are involved in the plasma membrane, the endoplasmic reticulum, and the sarcoplasmic reticulum. And those of you who are with me in nutrition, we talked about the hydrogen-potassium ATPase. This is the ATPase that allows the stomach to secrete hydrochloric acid, which we need in the digestion process. So again, P-type ATPases are classified as such because at least one of the subunits, they usually have two subunits, two identical alphas, two beta subunits, and at least one of the subunits is phosphorylated by ATP. This is something called primary active transport. So we'll talk about secondary active transport at the end. So primary active transport is pay me now, and secondary active transport is pay me later. So primary active transport means that when the transport process happens, that ATP is hydrolyzed, so you pay the price upfront. So the sodium-potassium ATPase, we've talked about this before, but here's a nice picture of what this looks like. And remember, what this does is it pumps 3 potassiums out and 2 sodium ions in, and both of them against the concentration gradients. And that's shown to maintain the high concentration of the potassium inside the cell and the low concentration of sodium outside the cell. So it is electrogenic. If you're pumping 3 sodiums out and 2 potassiums in, and they're both positively charged, then you are going to generate a charge there. It helps maintain cell volume through osmotic effects. And when the sodium ions come in, they bring water with them. So if that was allowed to take place unchecked, then the cell would eventually swell and then burst. It is interesting that of all the pathways we've talked about in Biochemistry 1 about how to generate ATP, for a lot of cells, about a third of cellular energy, that ATP is just used to fuel this pump. So I have a link to a video. I'm not going to play it here because I told you I get these echoey effects on my narration. So this is posted on Sakai, and it is I did actually post it to my YouTube channel to make sure that you were able to see it. So what this picture shows is the gradient between sodium and potassium. So potassium ions are pumped in and sodium ions are pumped out, and both of these are against the gradient. So the way that this works is here is our ATP, sodium potassium ATPase, and it's in its E1 conformation. And what happens is sodium ions have high affinity sites that they bind to, and the ATP binds as well. And then the ATP is hydrolyzed to phosphorylate the pump, and that causes a conformational change from E1 to E2. And then what happens is that changes the affinity of these binding sites. The sodium binding sites now become low affinity, and the potassium binding sites are now exposed and they become high affinity. So the potassium ions that are outside the cell are able to bind to their high affinity binding sites. The sodium ions are now low affinity, so they are able to leave. Here I point out that the phosphorylation is on an aspartate. So that's not shown in this picture right here, but the phosphorylation of an aspartate is what drives the conformation between E1 and E2. So the next thing that happens is that phosphate is hydrolyzed. So the hydrolysis of this phosphate then brings the conformation from E2 back to E1, and then now this makes the potassium binding sites low affinity, so they are released inside the cell, and now this creates the high affinity sodium binding sites, and the sodium is binding inside the cell on their way out of the cell, and the whole cycle repeats again. So I thought that you might find this interesting because there are a number of substances in nature that are sodium potassium ATPase pump inhibitors. So plant steroids. You can get these from, this is foxglove. So these cardiotonic steroids are derived from foxglove, and way back millennia ago, our ancestors didn't know how this worked, but they did know that foxglove could treat a weakened heart. Now we know how it works. So the digitalis compounds inhibit the pump, and that increases the sodium intracellular concentration. So that means that you have less of a sodium ion gradient inside and outside the cell. We have another exchanger, a sodium potassium exchanger. So without that large gradient, that means potassium ions are pumped out of the cell more slowly, and that means that the calcium ion concentration inside the cell will also increase, and that enhances the contractility of the cardiac muscle. So that helps strengthen the heart, and that's why these compounds are used to treat heart failure. So that brings us then to the calcium ATPase. So this is involved in primary active transport. It's a P-type ATPase. So as I mentioned before, calcium ions also often flood the cell, the cytosol, for a signaling event, but that means that the calcium has to be pumped back out of the cell after the signaling event is over. So in a resting cell, eukaryotic cells maintain very low concentrations of cytosolic calcium, maybe 10 to the minus 7th molar, and it might be 10 to the minus 3rd molar outside the cell. So a huge magnitude of difference here. So there are also calcium pumps in the ER and the sarcoplasmic reticulum, but the net movement in the resting cell is transport of calcium out of the cytosol. So it can go out of the cytosol by leaving the cell entirely, or it can be pumped into the endoplasmic reticulum or the sarcoplasmic reticulum if you're a muscle cell, and in that way the calcium also leaves the cytosol. So the way that this works is that 2 calcium ions are pumped out per cycle. So that's shown right here. So we have the stoichiometry isn't shown, but the calcium ions are pumped out of the cell. This is showing the calcium ATPase in the plasma membrane. And again, the pump is going to be phosphorylated, so we need ATP to do that. And then that keeps the cellular concentration of sodium very low in relationship to the concentration of the calcium outside of the cell. Here's the structure of the calcium pump, and you can see that it looks very similar to the sodium pump, and it does have these E1 and E2 conformations. The E1 is shown in purple and the E2 in green, so there's a pretty large conformational change that occurs when the pump is phosphorylated. So phosphorylation of the pump drives the conformation to the E2 conformation, and when that phosphate is hydrolyzed, you go back to the E1 conformation. So we do have calcium ATPase pumps in the sarcoplasmic reticulum as well. So if we orient ourselves here, if this is the inside of the sarcoplasmic reticulum and here's the cytosol, what happens is we have 2 sodium ions binding, and here we also have the involvement of hydrogen ions. So the hydrogen ions are going to be pumped. Um, when the calcium ions are pumped in, the hydrogen ions are pumped out. So this shows the binding of the calcium ions. The pump is phosphorylated. That drives a conformational change, and then that allows the calcium. Now we have low affinity calcium ion binding sites, so the calcium is released, whereas the um, uh, protons then that exposes a high affinity hydrogen binding site, so the hydrogen ions are pumped out. So this is a picture from our book right here. It shows the conformations of E1 and E2 and how that happens and what happens when that happens. But you notice in this picture there's no protein, there's no pump in this picture. So we should remember, that's why I have this picture right here, that there is a protein involved here. But these arrows show the different conformational changes that occur and how ATP phosphorylation and then dephosphorylation of the pump drives those changes. So some of you might remember in nutrition, here is another ATPase pump. This is the hydrogen potassium ATPase pump. Again, primary active transport, PATPase, P-type. So here we have chloride ions being pumped into the stomach. We should orient ourselves first here, sorry. So over here in blue, this is the inside of the stomach. These are the cells lining the stomach, and then this is the access to the bloodstream on the other side. So we have this chloride bicarbonate antiporter. We've seen this in lots and lots of different kinds of cells, but here the chlorides are moving in this direction and bicarbonate in that direction. So this is an antiporter where you're moving in opposite directions. The chloride then leaves the cell through this apical membrane. So this is the apical membrane on this side, and that's the basolateral membrane on the outside. We have these tight junctions that maintain the different identity of these membranes. So the chloride leaves these endothelial cells through this chloride channel. We have a potassium channel that allows potassium ions to leave, but then here is the hydrogen potassium ATPase, and what happens is the potassium ions are brought back in. So the way that this works is hydrogen ions are pumped out and potassium ions are pumped back in. So now here's our HCl. There's the H plus and there's the Cl minus, so that's the hydrochloric acid that's in our stomach. These hydrogen ions come from the dissociation of water into hydrogen ions and hydroxide ions, and the hydroxide ions then combine with CO2, and there's our favorite enzyme, carbonic anhydrase. So the hydroxide ions and the CO2 form bicarbonate, and then that helps complete the cycle so that bicarbonate ions are pumped out while chloride ions are pumped in through this antiporter. Okay, so those were examples of P-type ATPases. These are V-type ATPases. So these transport only protons. And I've alluded to in some of the earlier lectures that some of our organelles have a pH that's lower than neutral. So lysosomes, endosomes, plant vacuoles, the Golgi. So these hydrogen ATPases help maintain those pH differences. So this just shows a diagram of what the V-type ATPases look like. I was looking at this V-type ATPase, and it says that this is a vacuolar membrane, and maybe that's where that V comes from, but I've worked with a V-type ATPase before, and I was told that that means it was inhibited by vanadate. So I believe these V-type ATPases are inhibited by vanadate. So that's another thing that helps distinguish them from the P-type ATPases. So if we have our organelle right here, then the V-type ATPase is going to pump hydrogen ions in. And again, we're going to need the ATP hydrolysis because that will pump in the hydrogen ions against a concentration gradient. So now we have, for some organelles, that will also create an electric potential because you're pumping protons in. There are other organelles for which you don't want to do this, in which case you would use the V-type ATPase in conjunction with a chloride channel, and that means you're bringing in a proton along with a chloride ion. So there are some cases where you want that electrical potential, and there are some cases where you don't. So if you don't, you need the assistance of the chloride channel along with that V-type ATPase. Okay, what about F-type ATPases? Well, you have already studied this. Maybe you didn't know it was called an F-ATPase, but there's the ATP synthase that drives ATP synthesis, not hydrolysis, synthesis in the electron transport chain. So you might remember this from electron transport. Here are 3 alpha and beta subunits, and this is called F sub 1. And remember, each one of these betas synthesizes an ATP when this whole thing makes one complete turn. This F sub 0 is the stalk, and remember, it's the protons that are pumped out of the matrix and into the inner membrane space that come back in that turn this gamma subunit shaft right here that allows the rotation and the activation of the beta subunits during electron transport. So this is, one author calls this a reverse proton pump, because what it's doing is it's taking the protons pumped out through complexes 1, 3, and 4 in electron transport and bringing them back in, and that drives the synthesis of ADP to ATP, which is energy requiring. So those protons, that proton gradient that is established by electron transport is the energy reservoir that drives this ATP synthesis. So that's an F-type ATP synthase. Okay, so we remember we had 4 different categories. We had P-type, V-type, F-type, and this is the 4th one, the ABC superfamily. So this was first discovered in bacteria, but eukaryotic cells have them too. So this is called ABC because they have an ATP binding cassette. So that's what the AB stands for. So ATP binding leads to dimerization, whereas ATP hydrolysis allows these things to dissociate. Um, these are transporters of phospholipids, small lipophilic drugs, cholesterol, and other small molecules, where in the bacterial membrane, they might transport amino acids and sugars and peptides. So let's see how these things work. So this is a bacterial ABC, transporter, and this is used for the import and export of metabolites. So as the bacterial cells need them. So if we orient ourselves, if this is in the cytosol right here, then these are the ATPase domains that face the cytosol. So when they are bound to ATP, that causes conformational change that allows these small solute molecules to bind. But when the ATP is hydrolyzed, then the conformational change reverses, and then that allows this transported substance to be imported into the bacterial cell. So the way that works is that you might remember that bacteria have 2 membranes. So here's the cytosol. We have an inner membrane, and this is the periplasmic space. That's the outer membrane, and this is the cell exterior. So the way that this works is that the solutes come through the outer membrane in these porins, and these come through fairly easily. And then in this periplasmic space, there are binding proteins who are responsible for binding the proteins, and then they are responsible for delivering them to the ABC transporter. So that helps in the dimerization of this transporter along with the ATP binding, and then this allows the transported substance to be delivered to the cell. Okay, so that's what happens in bacterial cells. What about in eukaryotic cells? So in the eukaryotic cell, these are what's called MDR. Um, uh, um, this allows a cell to it's a multi-drug resistant protein. So in cancer cells, the cancer cells will overexpress these transporters, and this is bad if you are trying to kill the cancer cell, because that means that any drug you try to deliver to the cancer cell can just spit it right back out. So if we have our small solute molecule that, say it was a lipophilic drug that was able to cross into the membrane and enter into the cytosol of the cancer cell, it's able to bind to this ABC transporter. ATP binds, drives this conformational change, and ATP is hydrolyzed, sorry, and this drug is exported. So this is something that it's an adaptation that cancer cells have that allow them to export any kind of drug that you would use to try and kill that cell. So this, if the cancer cells are expressing these multi-drug resistance proteins, sorry, makes it really difficult to use your drug therapy to kill the cancer cells. I do have a video of this, so again, this is a narrated video, so I won't show it here, but it is posted on Sakai, so I encourage you to go and watch this. So these MDR proteins are members of the eukaryotic ABC superfamily because they work the same way. So they have these nucleotide binding domains right here, and that's where the ATP binds, and that drives the conformational change that allows the substance to be transported across the membrane. And again, overexpression of these makes them resistant to the cytotoxic drugs that would be used in cancer therapy. Okay, where are we? We're talking about transport across membranes using transport proteins, and we talked about ATP-powered pumps. Now let's talk about ion channels. Okay, so ion channels, these are involved in the nervous system, so I thought some of you would be interested in this. So there are three classes of membrane transport. We're now talking about ion channels. This is involved in passive transport. So that means that we are transporting our substance along a concentration gradient, not against a concentration gradient. So ATP is not involved here. So what happens is we have these ion channels that can be closed. They have a gate that would close them, but upon opening the gate, then the transported substance is just free to enter the cell through these channels. And again, there's sort of minimal interaction of the transported substance with the channel protein. But once the channel protein is open, these substances can come into the cell along a concentration gradient. So bacteria are able to make these ionophores. So these are small hydrophilic molecules. They dissolve in lipid bilayers and increase the permeability to ions, and they work in both directions. So they equilibrate concentrations of ions on either side of the cell. So this is another example of how bacteria have evolved to be able to they have these weapons against competitors. You know, restriction enzymes are an example of that. So mobile ion carriers and channel formers are examples of this. So a mobile ion carrier increases the permeability of membranes by binding an ion on one side of the membrane. And because, like their name suggests, it's a mobile ion carrier, it can bind an ion and then diffuse. And here we do sort of flip through the membrane, which we don't normally see. But these are mobile ion carriers, and it delivers the ion to the other side of the cell. So this would be a way for bacteria to use these to kill their competitors. And of course, molecular biologists, as we so often do, like with the restriction enzymes, we can use these for our own experiments. So valinomycin transports potassium. There is this compound called A23187 that transports calcium and magnesium. So what you can do is you can actually use these things as tools if you're trying to do an experiment where you want to deliver ions into your cells. So this is valinomycin. So this is a really interesting structure. So so this is what it looks like. And notice, because it's bacteria, they are very cleverly able to make D amino acids. So this structure is made right here. And this is a three-dimensional structure of what this thing looks like. And you can see this is the this is the potassium ion right here. And what happens is the carbonyl oxygens will form interactions between the to the potassium ion, which has a full positive charge. And this is the key to valinomycin being able to transport potassium only, is because of this octahedral coordination complex that it forms. So sodium and lithium, as we learned in general chemistry, are smaller than potassium. So sodium and lithium aren't able to form that coordination sphere. So sodium and lithium ions are not transported, only potassium ions are. Channel formers, so this is a channel forming ionophore. So they form pores, and this allows selected ions to diffuse. So gramicidin A, for example, transports monovalent cations. So this is what gramicidin A looks like. So again, these are, it's 15 amino acids, and again, because bacteria make them, we can alternate D and L amino acids. That's something we don't see in humans. And then it dimerizes in a head-to-toe fashion, and that forms the channel. So you form a helix that we don't normally see in mammalian cells. So it has a nonpolar exterior, so that way it's able to interact with the phospholipids of the membrane, and a polar central channel, and this one does allow both sodium and potassium ions to pass through. So this is a sort of a top view of what this looks like. And I do want to caution you that this is not a standard alpha helix. Standard alpha helices are formed of L amino acids only, and their cores are very tightly packed, and no ions could possibly get through. But when we have this dimer of this alternating L and D amino acids, we are able to form this pore in bacteria. Okay, where are we now? Transporters. Okay, so we have the three classes. We just talked about ATP-powered pumps, ion channels, and let's now talk about transporters. So transporters, as we've sort of already alluded to, we can put them in groups of either uniport, symport, or antiport. I do have a YouTube video posted, so again, it's narrated, so I'm not going to show it here. So uniport just means you have one substance transported in one direction. Symport are two substances transported in the same direction. Antiport means you have a substance two substances, one goes in one direction and one goes in the other direction. So we've already seen examples of this even just in this lecture. So if I go to my big table right here, I am here. So this is called facilitated transport, and facilitated just means you need a protein. So there's a plus sign right here, but we're transporting the solute along its gradient. So that means we don't need ATP hydrolysis. So the kinds of things that are transported in these proteins are glucose, amino acids, ions, and water. So let's look at some examples. And over here, so this is co-transport. So glucose, amino acids, and various ions and sucroses, these are antiporters. So there's a co-transporter. So let's look at I have examples of all of these. Okay, so transporters. There is a specific binding site to the specific solute to be transported, and then the transporter undergoes a conformational change to transfer that solute across the membrane. So here, this shows the solute binding. There's a specific binding site for that solute. There's a, we go through a phase where this is occluded, meaning it's not accessible to either the outside or the inside. And then there's a conformational change that occurs, and then that allows the solute to be transported to the other side of the membrane. And again, this is going along a concentration gradient, so no ATP is required. So I can show this animation here because this is just an animation, not a video, and there's no sound. I do need the Adobe Flash Player in order to do this. So let us talk about this. Oh, sorry about that. Let's get rid of that. Okay, it's nice to know this is secure. Okay, so this is a glucose transporter. So the glucose transporter binds to its transporter, and it binds very specifically. And Then we have a conformational change that occurs. We have this, that we sort of bring these subunits together up here, and these kind of open up on the other side. And then that allows the glucose to be delivered to the other side of the membrane. All right, so let's go back to our PowerPoint right here. So this is the structure of what this transporter really looks like, and this is a cryo-electron picture right here, and you can see there's the binding site for the glucose right there. So the glucose does bind very specifically. So again, it binds to the binding site. There's a conformational change that occurs, and that allows the glucose to dissociate. And then the conformational change reverts back to its original conformation, and that allows another glucose molecule to bind. So we have lots and lots of different glucose transporters. So this is GLUT1, so GLUT just means glucose transporter. And this just shows the same kind of thing. This is a different artist's interpretation. So again, we're taking the glucose along a concentration gradient. So glucose binds, there's a conformational change that allows the glucose to be delivered. But there's lots of different kinds of transporters. So this graph here shows GLUT1, which you find in red blood cells, and GLUT2 in liver cells. So, so those of you who are with me in nutrition, remember that all the glucose we eat that's absorbed in the cells lining the intestine, and then and then it enters the portal vein. So the portal vein goes right to the liver. So the liver gets the first crack at all the glucose that we eat. So in the portal vein, glucose concentration might be as high as 15 millimolar. So if you see over here, at 15 millimolar, the GLUT2 transporters will bind and transport the glucose. This diagram here means to show us what's happening, so that would be the fed state. But in the fasting state, fasting blood glucose concentration is 5 millimolar. So if we look at this graph at 5 millimolar, we can see that the GLUT1 transporter in red blood cells is transporting a whole lot more glucose than the liver. And that's good because the red blood cells really need that glucose. Remember, it's glucose, the liver's job is to act as a glucose buffer. So if you're fasting, the liver right now is going to be carrying out glycogen breakdown and gluconeogenesis. And the glucose transporter will be allowing glucose to leave the liver cell. And that means that the red blood cells can have it. So this differences in an affinity allow cells in a complex organism like ourselves to be able to get glucose to the cells that need it, and to be able to adapt in changes of fed and fasted states. So anyway, so this is what I would have asked you if we were in class. So if you look at the Km for GLUT2, you can see that the Km is about 5 millimolar. So for the GLUT1, okay, so this is if we look at the Km for GLUT1, it's a lot lower. It's like 1.5 millimolar. But for GLUT2, it's more like 5 millimolar. So again, with the Km of the GLUT2 being about 5 millimolar, that's about equal to the fasting concentration. And because the Km for GLUT1 is a lot lower than that, that preferentially allows the red blood cells to take up glucose during fasting conditions and not the liver. Okay, what about water? Okay, so aquaporins, they consist of 6 transmembrane alpha helices. I think this is really interesting right here, where you see the red and the blue, where this looks like a transmembrane alpha helix, but it's not. It's actually on 2 different domains of the protein. So they span only about halfway through the lipid bilayer, and that's shown over here too. So here it's orange and green, and these little spheres are water molecules. So aquaporins are tetramers, and in this diagram, the artist has used 4 different ways of visualizing protein structure to show how it works. So this is more of a cryo-electron microscopy picture here. These are the ribbon diagrams over here. One's in rainbow, one's just all in purple. And this is the space filling model right here. So you just see this little tiny pore where we can have our water molecules transport through. And here, this shows the amino acid side chains that are involved in the transport of the water molecules. So this is, I don't know if you can appreciate from here, but it's an hourglass type shape, and that's actually shown in our video. So I can link to this video because there is no sound, so I will go ahead and do that. So here are the water molecules, and they're sort of snaking their way through. And I'll show you in a minute, but there are amino acid side chains right here that force the water molecules to adopt a certain conformation as they go through the transporter. All right, so let's get out of there and go back to our PowerPoint. So what happens is we know that water molecules are bonded to each other by hydrogen bonds. So they need to break those hydrogen bonds so we can even consider water to have their own waters of hydration. And in the video, it was hard to see what amino acid side chains they are, but we have a histidine here, we have an arginine here, and then we have two asparagines right here. So it's really important then when we're transporting water molecules that we transport the water molecules one at a time, and I think you can see that in the video. But we have to make sure that we don't transport H plus or OH minus ions because that would change that they our text would cause us a proton conducting wire, and we don't want that. We just want neutral water molecules. So here, these green lines right here show the specific intermolecular forces that the water molecules are making with each of these amino acid side chains. And then these two asparagines right here, you can see that the water molecule, just one, is positioned between these two asparagines. And then that allows this one water molecule, and just one at a time, to be transported through this channel. So this is what a proton conducting wire would look like. This is from our Biochem 1 textbook, where protons can jump from one water molecule to the other. So that's what we don't want to happen here. We just want to transport one water molecule at a time. Okay, so up until now we've been talking about primary active transport, or if I go back. So primary active transport was the P-type, V-type, F-type, ABC superfamily. That was primary active transport. I want to define secondary active transport. So there are ion carriers that are linked to pumps like the sodium potassium ATPase. So what happens is you can have co-transport. So you can take something you want to transport against its concentration gradient, and you can have sodium come in with that thing you want to transport along its concentration gradient. So the sodium can bring in that other transported substance. But then you have to kick out the sodium, so then you have to use ATP there. So this is the pay me later. So the primary active transport was pay me now, and secondary active transport is pay me later. So the way that this could work, and those of you who are with me in nutrition might remember we talked about this, that the cells lining the intestine, some of them, the way that that they bring materials across, either amino acids from digestion, but here the example shows glucose. So if you want sodium and glucose to come across, really what you want is the glucose. But the glucose is coming in against its concentration gradient. So here they've drawn one glucose, and here they've drawn a lot of glucose. So the glucoses are coming in against its concentration gradient. The sodium ions are coming in, so there's a co-transport. The sodium ions are coming in along the concentration gradient. So you accomplish your goal by getting the glucose inside, but then the next thing that has to happen is then the sodium then has to leave, and you use the sodium potassium ATPase to do that, and of course you have to spend an ATP. So really you're spending that ATP to bring the glucose across. So there is another video that I will not show because this is a narrated video, but this shows how you can bring glucose across. So I believe we did talk about this in nutrition, but I can remind you. So here the intestinal lumen is over here on the left side, and we're wanting to bring glucose that's from our diet into these cells, the endothelial cells that constitute the brush border of our intestine, and we eventually want them to get to the portal vein. So here, glucose is brought in along a concentration gradient with sodium. So here the glucose is coming in against its concentration gradient, and sodium is coming in along its concentration gradient. But now what happens is the sodium it leaves, there's a sodium potassium ATPase right here where sodium exits and potassium comes in. So so now you maintain the sodium and potassium gradients between this cell and the blood. And meanwhile, there's a uniporter that allows glucose. So now glucose is going along its concentration gradient. So it came in against, and now it's leaving along, and now the glucose will enter the portal vein and will go straight to the liver. So that was our whirlwind tour of transport. I hope you enjoyed it, and I will see you next time.